So let's look at the big picture of how all of this happens. And to do that, we have to look at four major steps that must occur. First of all, there's events that occur at the neuromuscular junction. The muscle fiber is excited. Once it's excited, then there's what's called excitation contraction coupling, where basically the excitement of the muscle is paired with now the contraction of the muscle. Finally, there is the cross bridge cycle. So, what's really going to help you a lot that I definitely would recommend you to do is in mastering A&P, go to the study area. And in the study area, there is an area called A&P Flicks, and it should be the first three videos, but the videos are also directly linked to the ebook as well. The first one is on the neuromuscular junction, and they all do a really good job of showing these events happening. So this essentially is shown in that first video. The second video is called excitation, contraction, coupling. And it also does a really good job. So the excitation part happens in number 2 here, and it leads directly into number 3, which is excitation, contraction, coupling. And that would be video number 2. Finally, the third video that you'll need to watch is called the cross bridge cycle. So I'll be going over all of these in detail as well, but it doesn't hurt, especially for visual learners, which are the majority of learners in A&P, to see these really well done videos also. So for the big picture, I'm going to zoom in, then I'll zoom back out. But first we see the axon of the motor neuron. So what we have here is what's called the motor neuron, and the definition of a motor neuron is going to be the axon specifically that excites the skeletal muscle. So that's why we use the term motor here. So that's an important term to make sure that you know. So the first thing is there is an action potential that travels all the way down the axon, which is the first thing that is shown right here. So I'm going to represent this action potential by an arrow that's jumping down the axon all the way until it eventually reaches the end of the axon, which is called the axon terminal, which we've looked at. So the motor neuron's axon terminal then releases acetylcholine into the synaptic cleft. Remember, the acetylcholine is in a synaptic vesicle. It's released by what's called exocytosis, which you learned about earlier on in the semester. The acetylcholine binds to its receptor on the junctional folds of the sarcolemma. The acetylcholine then binds to cause what's called a local depolarization. And this local depolarization is not an action potential. It's what's called a graded potential, and the graded potential occurs because there is sodium goes in and potassium goes out at the same time. So it's weaker than an action potential. So we have sodium going in and potassium going out simultaneously. So that makes it different than an action potential. And in this unit, in chapter 11, you're going to learn all about the differences between a graded potential and an action potential. Now, this graded potential leads to an action potential. So it starts out as a graded potential, but it becomes an action potential. And that's the beginning of the excitation-contraction coupling. So the first part here is, again, in that first video in A&P Flicks. The second video is shown here. And remember that the sarcolemma is a plasma membrane because it can conduct an action potential. And it extends into the T tubules, which are an extension of the sarcolemma. So it essentially is a plasma membrane as well. So we have this action potential continuing down into the T tubules. As a result of that, it causes calcium to be released from storage, which remember is in the sarcoplasmic reticulum. Calcium binds to troponin, which shifts the tropomyosin to uncover the myosin binding site on actin. So during this period, there's really two important chemicals that are super, super important. There is calcium that's important. There's also ATP. The calcium is released from the sarcoplasmic reticulum, and it binds to troponin. The ATP is broken down by the myosin. And remember, the myosin is an enzyme, which is ATP ACE enzyme, breaking ATP down into ADP. So finally, the cross bridge can occur. And the cross bridge is video number 3 in the A&P Flicks that you're going to watch. And what happens essentially is that the myosin grabs the actin and pulls the actin towards the H zone, and the entire sarcomere actually shortens. So there was a whole lot on that slide, so feel free to pause the video, go back to it, take a screenshot, whatever works. So now we're just going to look solely again at the neuromuscular junction in this slide. So you should know these steps. You're not going to have to list them or anything, but you should be able to pick out from a multiple choice question what the proper order is. So we have action potential arriving at the neuromuscular junction. The next thing is voltage calcium channels open. Calcium enters into the axon terminal. Calcium is what causes the release of the neurotransmitter. So the synaptic vesicles move to the end of the axon terminal and are released by exocytosis. The acetylcholine diffuses across the synaptic cleft, binding to the gates on the sarcolemma, and that causes sodium to go in and potassium to go out, which is a local potential called a graded potential. So it has the end plate potential, which again is this graded potential, but the graded potential leads to an action potential, which starts to travel down the sarcolemma into the T tubule. But after the acetylcholine's done its job, this is a very important step. There is a special enzyme called acetylcholine esterase, and you don't have to write out the whole thing because it is confusing, but acetylcholine esterase, as we can see here, is one way that a neurotransmitter can be removed from the synaptic cleft. This enzyme is going to break acetylcholine down into its acetyl part and its choline part, so that it can no longer have an effect at the synaptic cleft.